Okay, so welcome to this next video in which we're discussing the opening mechanism of cis-loop ligand-gated ion channels. Okay, right, so we've seen the example in the case of the nicotinic acetylcholine receptor, but we'll now generalize out again. This will become important so that we can talk about the difference between receptors which just have two ligand binding domains and those which have 5. But actually this principle of having the ligand binding domain in between two of the receptor subunits like this is common to all of the cis-loop ligand-gated ion channels. So basically what we'll do is we'll talk about this in more generality. So if you have one receptor subunit here, so this is one of the receptor subunits, and then you have its neighbouring one here, and they're forming a ligand binding site in between them. And we're not now thinking of this just as being acetylcholine anymore, we're thinking of it being as a more general ligand, such as GABA, serotonin, glycine. Okay, so this is our ligand binding site. Then what you find, basically, is that one of the subunits that makes up a wall of this ligand binding site provides certain motifs into this ligand binding site. So basically, we can see that the ligand binding site is in the cavity between two of these receptor subunits, basically. Now, that means that the walls of this cavity are lined by the protein. Now, what sort of portions of the protein does the protein stick into the cavity? What motifs does it have sticking into the cavity? Well, basically, one of the proteins which lines the wall of this cavity will have 3 loops, okay, which I'll draw here, known as the A loop, the B loop, and the C loop. Okay, so all of these are loops. A, B, C loops. Okay, and the subunit which provides these A, B, and C loops into this cavity that's going to form the ligand binding site, this is what's known as the principal subunit. So this is the principal subunit. Principal, I think it's LE like that. Principal subunit. I think the other way is referring to like a head teacher. Right, so principal subunit. Then the other subunit provides 3 other motifs which are also referred to as loops, even though they're not actually loops. Instead, they're beta-pleated sheets. So you have the D, E, and F motifs, which are still called the D, E, and F loops. However, they are not actually loops. The A, B, and C motifs were actually loops. The D, E, and F loops are actually beta-pleated sheets. But the nomenclature for these is still to call them the D, E, and F loops. So these are beta-pleated sheets. Okay, right. So, the subunit which provides these D, E, and F motifs into the ligand binding site is then known as the complementary subunit. So for every single ligand binding site, you have one subunit which is acting as the complementary subunit and one which is acting as the principal subunit. Okay, and they together form this ligand binding site. Right, so what we now want to discuss then is how does the channel actually open. So basically, when the ligand binds to the ligand binding site, okay, what's going to happen is this C loop here is then going to close. It's going to change conformation and it's going to close over the ligand binding site. So let me draw this. Let me turn over and draw this. So what's going to happen is if we have our we'll take the example of the skeletal muscle nicotinic acetylcholine receptor. So here it is. Here is its receptor subunit composition. So remember, this is the alpha-1 to beta-1 delta epsilon receptor. So we'll assume that it's the adult form rather than the fetal form. So epsilon there, alpha 1 here, beta 1 up there, and then delta down here. And we know that the two ligand binding sites are here and here. So when ligand actually binds to them, so we'll show this in pink here, what's going to happen is the C-loops, which by the way are provided by the alpha 1 subunit. So the alpha 1 subunit here is acting as the principal subunit, and the delta and the epsilon subunits are acting as the complementary subunits. So the alpha 1 subunits will have their C-loops change conformation so that they now cover up this ligand binding domain here. So they're going to fold across the ligand binding domain. Now what that is going to cause is it's going to cause a movement in the whole alpha 1 subunit. Okay, and what's going to happen is the alpha 1 subunit is going to twist, basically. They're both going to twist in this sort of direction. Okay, now remember the structure of these subunits. So what you have is you have this extracellular domain, which is the portion involved in all of this ligand binding domain. You then have the M1 membrane spanning alpha helix, the M2, the M3, and whoops, I've forgotten the intracellular loop, almost forgot it there. And here's the C-terminus of our protein. So here's the amino terminus up here, and then we've got the cell membrane here, which the protein is sitting in. So what's going to happen is this is going to twist, and it's going to twist the M1 helix, which is then going to twist the M2 helix. So what's going to happen is, if I draw another picture, okay, so in this picture I'll show these M1 and M2 helices. Okay, so this is going to be a picture of the transmembrane region of this skeletal muscle receptor, rather than the extracellular region. So we know that the pore is formed by these M2 membrane spanning alpha helices here. Then outward from that is the M1 membrane spanning alpha helix here. Okay, then we've got M3 and then M4. So I'll color code these again for you. Right. Okay, so M3 here, M4 here, M3 here, M4 here. So let's color code this picture again, otherwise it's a total nightmare if you don't. So this is the M2 membrane spanning alpha helix, which is the one that's actually lining all of these pores here. Right. Then we have the M1 membrane spanning alpha helix, which is the one which is going to be attached to the extracellular portion. So when the extracellular portion moves in response to the ligand binding, it's going to be the one that feels that movement first. Okay, so the M1 is bound to the M2, like so, by this loop here. Then we have the M3 membrane spanning alpha helix, which we'll have in green here. Okay, there and there. So this is this one here, which is again linked to M2. Okay, so these two are linked, like so. And then finally you have the M4 membrane spanning helix, which is here, which is then linked off the M3 membrane spanning helix, like so. Now basically, what's going to happen when ligand binds, it's going to cause this change in the conformation of this C-loop, and it's going to twist this way. That's going to cause both the M1 and the M2 loops to also twist this way. So all, well, the two ones which have the C-loops on are going to twist anyway. So these two, sorry, just these two alpha 1 subunits. So this is an alpha 1 subunit and this is an alpha 1 subunit. Because remember, we've only got two ligand binding sites here. However, when these two twist, it pushes all the others. So all the others twist as well. So the others do twist as well, but it's not because they themselves have got this ligand binding portion happening. So these are going to twist like this as well. And finally, these ones are also going to twist this direction. So everything's going to basically twist like this way. So everything's going to twist anti-clockwise. Okay, right. Now, the fancy nomenclature for this twisting is that we say that the inner leaflet of the transmembrane region is twisting. So inner leaflet. So let me tell you what we mean by the inner leaflet of the receptor. We mean the portion that is closest to the pore of the channel. So basically the M2 alpha helix and also maybe the M1 you would call the inner leaflet. So what you definitely don't mean is M4 all the way out here. That would be the outer leaflet. So the inner portions of the channel is the inner leaflet and the outer portions would be the outer leaflet. So the point is that what you've got happening is all of these M2 alpha helices are twisting a little bit. And when they twist like this, the way that the M1 and the M2 alpha helices are oriented with respect to the ligand binding domain changes, and basically the whole thing relaxes a little bit, and all of these M2 alpha helices relax. So basically what ends up happening is they all move outwards, like so. So they all open the pore, basically. They widen the pore. So all of the M2 alpha helices move outwards, basically, to widen this pore. And that's known as M2 alpha helix relaxation. Now, what's going to happen when you relax the alpha helices? Well, remember, all these M2 alpha helices have those hydrophobic amino acids coming off their sides. So I'll show these in orange. They were either leucine or valines of the 9' amino acid residue of the M2 alpha helix. So all of those are going to move outwards, and basically you're going to open a gap in between through which ions can move. So relaxation is going to lead to the opening of the hydrophobic girdle, opens the hydrophobic girdle, and that means that ions can now move through the pore of the channel because they're not going to have to interact with the hydrophobic residues at that 9' position. Okay, so that overall is how ligand binding to the ligand binding domain is going to change the conformation of the transmembrane region to pull the hydrophobic amino acids at these 9' positions, these hydrophobic girdle, outwards and open up a pore through which ions can move.